Welcome everyone. Today I'm going to discuss about genetic engineering, and this is very important for our discussion. Genetic engineering, it's a biotechnology. We have been manipulate DNA for generations. Artificial breedings, creating new breeding cells and new crop plants to improve our food. Animal breedings, bred for food plant. Descendants of the wild mustards, the cabbage family. Breeding food plants, evolutions of modern corn from ancestral Tessunians. A brave new worlds. Victim response seems suspect blood. Victim rape seems suspect blood. The code is universal since all living organs use the same DNA, use the same code book, read their genes the same way. Human genomes 3.2 billion base. Can we mix genes from one creator to another? Yes. Mixing genes for medicine, allowing organisms to produce new proteins. Bacteria producing human insulin, bacteria production human growth hormone. How do we mix gene? Genetic engineering. Find gene, cut DNA in both organisms, paste gene from one creators into other creators' DNA, insert new chromosome into organism. Organism copies new genes as if it were its own. Organism read gene as if it were its own. Organism produce new protein. Remember, we all use same genetic code. Cutting DNA. DNA scissors and enzymes that cut DNA restricts enzymes used by bacteria to cut up DNA of attaching a virus. E. coli, Hindil, Bemhi cut DNA at specific sites. Enzyme look for a specific base sequence. Restriction enzymes cut DNA at specific sites. Leave strictly ends strict enzyme cut site. Cut other DNA with same enzymes, leave strictly ends of both, cut glue DNA together at strictly end. Gene you want, chromosome want to add genes, combine DNA. Strictly ends help glue genes together. Cut sites, gene you want, cut sites. Strictly end isolate genes, cut sites, chromosome want to add genes. DNA laser join the standard strictly end sticks together. Recommendation DNA molecules, chromosome with new DNA added. Why mix generation together? Gene produce protein in different organism of different individuals. Human insulin gene is bacteria. New protein from organism, example human insulin from bacteria. Uses of genetic engineering. Genetically modified organism enabling plant to produce new protein, protect crop from insect bat corpse. Corn produce a bacteria toxic that kills corn's borer, extend growing seasons. Fish bearer, straw borrower with an anti-freezing gene from flounder. Improve quality of food. Golden rice producing vitamin A, improve nutritional value. Bacteria are great one call organism, reproduce quickly, mitosis, easy to grow, fast to grow, generations every 20 minutes. Bacteria DNA, single circular chromosome, only one copy is equal to haploids, no nuclear, one DNA, plasmids, bacteria chromosome. There are more plasmids, small extra circles of DNA carry extra genes that bacteria can use, can be swapped between bacteria, bacterial sex, rapid revolution, antibiotic resistance can be picked up from environment. How can plasmid help us? A way to get genes into bacteria easily, insert new genes into plasmids, insert plasmid into bacteria. Vector bacteria new express new genes, bacteria make new protein. Grow bacteria make more. Genes from one organism recombines plasma, transforms bacteria, grow bacteria, harvest protein. Applied of biotechnology, bacteria, bacterial plasma, isolation plasma, gene insert into plasma, cell contain genes of inserts, plasma put bacteria, cell phones joins in insert, identification of deserts cone, copies on genes, copies on protein, various applications. These are the different kind of applications of the genes, and this is very much important for us. And if we focus on some research paper on genetic engineering, we find that there are many things many things we are actually utilizing. So if we do some sort of genetic research which will help us to grow our economy in our effective way. So if we can understand the genetics engineering, if we can understand the genetics things, if we can understand the mm, uh, different kind of important aspects, then it will be very much effective for us. And most important thing is that the more we can understand the genetics, we can understand different kind of factors. Especially I provide the application of the biotechnology, and through this application you can understand the economy. Through this biotechnology you can understand the process and everything. So today I just shared one of the research paper. This research paper is very much important. Especially this research paper can help us to grow our economy in an effective way. If we can understand these things, low frequency variant associations with leukoid telemounts length in Singapore Chinese populations. This is Zhuang Lianchang, Reshma L. Gurungs, Rajkumar Doraju. They are the main scientist researcher. Abstract. The role of low frequency variant associated with telomere length homostains in chronic disease and mortalities in relatively understand in the East Asian population. Here we evaluate low frequency variant including 1915154 Asian specific variant. For leukocyte telomere length associations among 25533 Singapore Chinese samples, three station specific variants and near POT1 and other goals are associated with LTL. A massive variance shows effective 5.3-fold higher and independence of the previous common index SNP. TARF F1s and STN are linked to LTL associated common index SPN at this losses. This is associated with cancer mortalities and of associated between RS and colon cancer is mediated through LTL. Overall, genetically determined LTL is particularly associated with lung adenocarcinoma. Ethically specific low frequency variants may affect LTL homostasis and associated with certain cancers. So these are the way we can understand the genetics low frequency variant associated with leukocyte telomere length in the Singapore Chinese populations. These are the different kind of variant we can understand. This is the way we can understand the genetics engineering. Genetics engineering can understand the DNA and DNA factors and how we can regenerate this DNA in an effective way. This is also important. So different medical science, different kind of biotechnology, different kind of important technology, different kind of factors. We have to understand that we must need to be applied this kind of genetics things in an effective way. And if we can apply this kind of things in an effective way, that time we can take necessary initiative and we can move with that effective way. So thank you everyone. If you have any kind of question regarding genetics engineering, if you have any kind of plan regarding genetics engineering, this will be very much effective, and this way we can grow together, we can understand together, and we can develop our things in an effective way. So thank you everyone. If you have any question, you can directly communicate with us, and I can give you the answer how actually genetical engineering may work and how it will be helpful in the COVID-19 situation. Besides that, we are running World Talent Economy Forum. The World Talent Economy Forum actually is a high qualified think tank, researchers, organizations. They are working with World Talent Economy Forums, and we can promote very effective discussion in an effective way. Thank you, everyone.